Hi, my name is Matt Durham. I did my summer research at the University of Michigan's Chemical Engineering Department with Professor Savage, and my summer project was algal growth and biodiesel production. There were two main things we did in the lab. The first was algae production, and we grew the algae with different carbon nitrogen sources to try and figure out what was best to grow the most algae and also to maximize the oil in the algae, because that's what was later converted into the biodiesel. And the second step was converting the algae into biodiesel, and this was done by reacting the algae under high pressure and temperatures. And you can see in the bottom corner here is a picture of one of the members in our lab holding a vial of the biodiesel product. This is just a chart that shows oil yield of different crops that are grown for biodiesel. And you can see that microalgae is at least 10 times higher than the rest of the crops. And you also notice that there are two different numbers for microalgae, and this is due to the fact that under different growing conditions and different species of algae have different amounts of oil within them. When we grew the algae, there were two main stages we had. The first was the photoautotrophic conditions. This means that it was grown in light and it underwent photosynthesis and grew. And the second stage, we grew it in either photoheterotrophic conditions or chemoheterotrophic conditions. Photoheterotrophic conditions mean that we gave it light and a carbon source, such as glycerol and glucose. And chemoheterotrophic conditions means we did not give it any light, so it was in the dark with a carbon source like glycerol or glucose. And then in addition to these stages, separately, we had two different phases that we had for growing. The first stage, we gave it lots of nitrogen, and we wanted the cells to grow quickly and divide. In the second stage, we gave it all the nutrients it needed except for nitrogen, so that the cells couldn't divide, but they would grow larger and they would get more oil in them. When we did the hydrothermal reaction to convert the algae into biodiesel, there's an aqueous phase that we recover that contains carbon and nitrogen. And this is useful to recycle and reuse those nutrients and grow the algae off the carbon and nitrogen so we can just have a closed cycle and recycle these nutrients. But first we had to make sure that these nutrients were exactly what it needed. So we wanted to make sure it could grow on the ammonium, which is what's contained in the aqueous phase. So we tested out different nitrogen sources of ammonium and nitrate. And we found that when you grow algae on ammonium, it lowers the pH a lot, so much that it is so acidic it can't grow. But to fix this, all we had to do was just add a little base and keep the pH around 7, and it grew just fine. So the algae can grow on either ammonium or nitrate. And we also wanted to check out the carbon sources. When algae grow, it'll take a simple sugar that you give it, like glycerol or glucose, and it can break that down and use the energy to keep growing. And if you don't give it a carbon source, like a sugar, then it'll also just use the CO2 that's dissolved in the water and use that for the energy during photosynthesis. Since glycerol was contained in the aqueous phase, that's what we wanted to make sure it could grow on. And another reason we checked glycerol is that it's a byproduct of the reaction to produce biodiesel. So we have a lot of crude glycerol left over that we want to use somewhere. And we did find that the algae is capable of growing on the glycerol. So in conclusion, growing the algae in two stages is best to maximize the amount of oil we can get with high nitrogen first to divide the cells, and then low nitrogen so the cells grow larger. And then we have lots of opportunities to recycle waste products, and we need to do this to A, be more efficient, and B, it's going to help with the life cycle assessment if we can keep it a closed loop and recycle this aqueous phase in crude glycerol. Thank you for your time.